Hi, everyone. My name is Peter Nguyen, and I'm a junior undergraduate student in the Microbiology Department at UMass Amherst. Here at UMass, I'm a member of Dr. Morita's lab, and today I'll be presenting my research. My project is titled Effect of Membrane Stress on Phosphatidylinositol Mannosides in Mycobacterium smegmatis. Mycobacterium are a genus that contain multiple pathogenic agents that are deadly to humans. One of the most notable ones is called Mycobacterium tuberculosis, which causes tuberculosis. This is also known as TB. TB is a very deadly disease that remains the leading cause of death from a single infectious agent. In 2018, 1.2 million people died from TB, while 10 million people were infected worldwide. Recently, multi-drug resistant strains have developed, making it very difficult to successfully treat infected individuals and also making eradication efforts very strenuous. Scientists from all around the world are currently researching novel drug targets within the organism to combat drug resistance and to also improve treatment of tuberculosis. One of the current drug targets is plasma membrane and cell wall biosynthesis within the mycobacterial cell. The plasma membrane and cell wall make up the cell envelope, and one of the main issues is that the assembly and functions of the cell envelope is still not fully understood. The Morita Lab is currently researching many aspects of cell envelope synthesis to improve our understanding of mycobacteria. At the lab, we mainly use a model organism called Mycobacterium smegmatis, which is non-pathogenic, so it's a lot safer and easier to work with. Within the plasma membrane is a glycolipid biosynthesis pathway, which produces phosphatidylinositol anchor molecules like PIMs. My project focuses on PIMs, and we care about them because previous research indicates that these membrane-associated lipids are important for growth and viability in mycobacteria. PIM is short for phosphatidylinositol mannoside and is generally composed of phosphate, inositol, mannoses, and acyl chains. One example of a PIM is shown here, which is diacyl PIM2. The AC2 represents the two acyl chains, the M2 represents two mannoses attached to the inositol, and PI represents the inositol, phosphate, and the diacyl group attached. When we use the term diacyl, it refers to the two fatty acids in addition to the diacyl PIM. In terms of location, PIMs are primarily found in the plasma membrane, with some being found in the outer membrane as well. Here's the PIM biosynthesis pathway. It begins with PI, or phosphatidylinositol. A mannose transferase mediates the transfer of a mannose, which makes PI become PIM1. As we go further down the pathway, you can see that PIM1 can have additional mannoses transferred to make higher forms of PIMs, such as ACPIM6. In addition, acyl transferases can add an acyl group to form higher forms of PIM, such as ACPIM2 and ACPIM6. In addition, ACPIM4 can be further monosylated to become lipomannan, or LM, and lipoarabinomannan, which is LAM. These are other important glycolipids in mycobacteria, but we won't be focusing on them today. So ACPIM2 and ACPIM6 are the predominant forms of PIMs, while diacyl PIMs such as diacyl PIM2 and diacyl PIM6 are secondary products produced in smaller amounts. And it was found that during membrane stress caused by treatment of cells with benzyl alcohol, a membrane fluidizer, diacyl PIMs become dominant compared to monoacyl PIMs. This suggests that an acyl chain is being added to the inositol of ACPIM2 and ACPIM6. The structure of benzyl alcohol is located in this image right here. Because of this finding, I hypothesized that inositol acylation of monoacyl PIMs is a response to membrane disruption. In cells, the addition of an acyl group is potentially a way to rigidify the membrane in response to an increase in membrane fluidity, since benzyl alcohol fluidizes the membrane. My main objective for this project is to explore PIM acylation mechanisms by inducing membrane stress. So to do this, I induced membrane stress using various methods, such as treatment with chemical compounds, heat stress, and genetic approaches. So here's just a brief overview showing how we extract and analyze PIMS from M. smegmatis cells. We grow the cells in a flask, and then we spin down to a cell pellet. We then use organic solvent to extract lipids. Afterward, we use thin layer chromatography, or TLC, which separates components based on polarity. This allows for separation of different PIM species. Afterward, we stain with the compound, like orsinol, by spraying, and then this allows for us to visualize the PIMS on the TLC plate. In this experiment, I treated M. smegmatis cells with benzyl alcohol, or BA, at a concentration of 100 millimolar for 1 hour to see the difference between non-treated cells and BA-treated cells. I'd also like to note that DMSO is the vehicle control since it's used to dilute BA before treatment. So in this TLC image, we can see that in non-treated cells and DMSO-treated cells, there's predominantly ACPIM2 and ACPIM6 as expected. But upon addition of BA, diacylpim2 and diacylpim6 accumulate, while the monoacylpims deplete in some sort of PIM ratio flip. This ratio switch indicates that inositol acylation of monoacylpims to form diacylpims occurs as a response to BA. And because of this, I suspect that this response is a modification of existing PIMs rather than new PIMs being synthesized. To determine how long acylation takes to occur, I treated cells with BA and then extracted lipids at different times up to 40 minutes. And as we see on this TLC plate, acylation occurs within 20 minutes. This fast response suggests that inositol acylation is an immediate enzymatic response rather than a consequence of signal transduction and gene transcription. To prove that accumulation of diacylpims is a biological process rather than being due to the fact that benzyl alcohol might be somehow improving lipid extraction efficiency, cells were killed instantly using microwave radiation and then afterward treated with BA. As you can see in this TLC plate, there's no acylation occurring when cells are dead, even after treatment with BA. And you can see that on the right of the TLC plate, there's no difference in PIM profiles between both microwave samples. Cells from this entire experiment were also serially diluted and then plated onto agar to confirm that cells are dead after microwave treatments and not dead after the other two treatments. Another way to alter membrane fluidity is to use heat. It's known that high heat can increase membrane fluidity, so I heat shock cells at 55 degrees Celsius in a water bath for up to 30 minutes. Even after just 5 minutes, there's an accumulation of diacylpims, and this result is very similar to that of BA treatment. And just like before, cells were diluted and then plated onto agar to confirm that they're alive and viable during heat treatment. To summarize, benzyl alcohol, a membrane fluidizer, can induce PIM acylation. The addition of an acyl chain to ACPIM2 and ACPIM6 is a biological process. This means that benzyl alcohol is not affecting lipid extraction efficiency. And lastly, membrane fluidization due to heat stress can induce inositol acylation. For the future of my project, I plan to use genetic means to alter the membrane rather than chemical treatment or heat stress. The two genes I plan to knock out encode for DESA3 and CFA, both of which play a role in membrane fluidity regulation. DESA3 is a fatty acid desaturase that adds a double bond to saturated fatty acids, which creates unsaturated fatty acids. Doing so would increase membrane fluidity. CFA is a cyclopropane fatty acyl phospholipid synthase, which acts on the double bond in unsaturated fatty acids to create cyclopropane derivative within the acyl chain. So it becomes cyclopropanated fatty acid. So this would make the plasma membrane more rigid. The goal is to see how inositol acylation of PIMS would be affected after knocking out these genes to alter membrane fluidity. And that concludes my presentation. Thanks for watching, everyone.